Thank you. Good morning to everyone. Squalo Normale is coordinator of Spoke 8 in the project. And I will be talking to you today about work that we are doing in my own group. So, scusa. Okay. So, let me define what is a nanobody. So, on the left there is an IgG that has variable regions and constant regions, and these are the standard way how therapeutic antibodies are used. Nanobody is a smaller portion of the antibody that encompasses only the two variable regions in the form of a single chain Fv fragment, or VHH, only heavy chain antibodies. So my definition of nanobodies will be that either a VHVL, heavy and light chain, or a VHOVL single domain antibodies. And what I'll be talking to you about is how we have pioneered the technology over the many, many years to use antibodies as genes. Normally, therapeutic antibodies are used as proteins, and the therapeutic antibody market is the largest market of biopharmaceuticals, as has been mentioned before. And however, therapeutic antibodies by their format are limited to the world of extracellular targets, because you cannot have an antibody to access the intracellular portion of a cell in any tissue. And the intracellular environment is a huge source of disease targets. So our idea over the years has been, can we harness the power of antibody repertoires to hit intracellular targets? And so this is the main summary of work that we have been doing for many, many years. And the example that I will give you today is in application of this approach in Alzheimer's disease. And I thank my previous speakers, the previous speakers, to have made the introductions to the. So currently, Alzheimer's disease has a main extracellular view. So this is a dominating paradigm of Alzheimer's disease, a prion-like transcellular spreading of misfolded proteins, whereby vulnerable neurons start making a misfolded protein. This then is targeted, is exported outside the extracellular environment, and it hits neighboring cells. So like in a fire hitting a forest, the fire propagates and the neurodegeneration propagates. And the idea of therapeutic antibodies, as in the classical format as are used in Alzheimer's, being developed in the field for the past 25 years for Alzheimer's disease, is to intercept the misfolded protein in the extracellular environment. However, we know that the efficacy is rather limited, unfortunately, for these therapeutic antibodies, and also there are many side effects. So this is really the problem in the use of therapeutic antibodies in Alzheimer's disease. And so this is. There is also another point, that this is a cartoon that you will find in any article on amyloid, and the cleavage of amyloid beta peptide is depicted as happening directly in the extracellular environment. This may well happen from the membrane APP, but as we have demonstrated, and as I will show you, we have demonstrated, and others have demonstrated, that the first production of A-beta occurs inside the neuron. So this is where really you want to hit the cell autonomous phase of neurodegeneration. And even more so, tau. Tau is an intracellular protein. It first misfolds intracellularly, and then it is, when the neuron degenerates, it is found extracellularly. So we really want to hit misfolded proteins inside the neuron. And so we want to hit the first cell autonomous damage to neurons where it is first done. And how to do this? And so we have developed this concept first, and then we have developed libraries, a platform of intracellular antibodies, intrabodies as we call them. They are intracellular nanobodies. And we can harness the power of the fact that we can derive from our libraries conformational selective and post-translational specific nanobodies against the misfolded protein. So this is what I will briefly tell you today now. And so the idea is that you have a gene that encodes for a specific nanobody, and you can target it to different intracellular compartments, in cytoplasm, nucleus, mitochondria, ER, membrane, synapse. And so that is really the power of how you can achieve subcellular precision coupled to molecular precision that the nanobody provides. And so this is the whole idea of the platform that we have been developing. And of course, the gene-delivered antibody is made by three moieties. The first moiety is the intrabody itself, so it has a specific binding to an intracellular target that can be post-translational modification specific or conformational specific. Then you equip this with a subcellular targeting sequence that will direct the gene after it is translated to a specific compartment so that you can really be subcellularly precise. And then one approach, you can equip the intrabody with an effector domain, for instance, a Degron domain that will target the intrabody to be for targeted degradation of the recognized antigen. And so this is the idea, targeted degradation of misfolded protein inside neurons with molecularly precise nanobodies. And so we have libraries that we have developed that from which we call these libraries, splint libraries, single pot library of intracellular antibodies, whereby we can select by intracellular antibody capture technology a nanobody directly from the gene sequence that encodes for the antigen. So in our libraries, unlike any other technology that is available for making human antibodies, our technology, you have, I don't have the time to give you the detail, but the idea is that you have your target, your antigen is a cDNA that is encoded by a yeast cell, and then you have your antibody library, and the system is such that if an antibody in your library, or a nanobody in your library actually, binds to the antigen that all cells in your library express, this cell will survive, whereas the other cells will not survive. So you set up the system, you go home, 2 weeks later you come, and surviving cells will have the antibody that binds your antigen. Now, this is making it easy, but this is exactly from cDNA to antibody. So this accelerates enormously the process of antibody generation. And so these are our libraries, and we have libraries from naive libraries, so these are general libraries. We have them from camelids, from mice, from humans. We have patient-derived libraries, and so we can provide also patient-specific antibody libraries. And so we can go from DNA to nanobodies, no need to immunize animals, no need to produce and purify the antigen. We can do affinity maturation if necessary, and this is fast and scalable. So this is the first asset that our future startup company is going to build on. And second point is we have made the variant of these libraries to derive antibodies against post-translationally modified proteins. So these are, on one hand, a very difficult target because having a drug, no FDA-approved drug targets the post-translationally modified version of a target. On the other hand, they can be very, very disease relevant, and they are an enormous source of targets. So how can we derive an antibody against a post-translationally modified protein in the native conformation? We made we developed this platform that is called PISA, post-translational intracellular silencing antibodies. We have a granted patent on this technology. And so PISA platform is a discovery engine. We have proprietary libraries that can hit post-translationally or conformational proteins in a native conformation. We can translate them inside neurons or cells and target them to different compartments. And then these now are optimized for functional interference, for instance, targeted degradation. And what we are doing this now is in our Square Normale spin-off company, we will leverage the discovery platform for a new generation of drugs that are these nanobodies against the target proteins that are conformational specific, such as the beta oligomers, or post-translationally modified proteins, such as, for instance, phospho-tau. I will give you rapidly 2 examples. And so the drug candidates derived from the platform will be developed up to the end of regulatory preclinical studies, then to be licensed out. And so remind that I told you, I hope I convinced you that the first cell autonomous damage to neuron is done intracellularly. So both A-beta and tau are first formed intracellularly, they aggregate intracellularly, and then they are exported outside. And so the first product that we are developing in the lab is a nanobody against phospho-tau. You know that when tau is abnormally phosphorylated, it detaches from microtubules, it aggregates in the cytoplasm, and this is where tangles are formed. So instead of targeting whole tau, that is a physiological normal protein, we want to target the phosphorylated version of tau and target specific phospho-tau, but not normal tau, to degradation. This is the first, we have an active program in the lab working on this. And the second approach is to target A-beta oligomers, specifically that we have developed by Giovanni Meli and my group over already 10 or 12 years ago. And these nanobodies specific for A-beta oligomers, we have demonstrated that both in cells and in vivo in mice, we can use them, first of all, to demonstrate formally that when we express this oligomer-specific intrabody in the endoplasmic reticulum of a neuron, there the oligomers are first formed, intracellular oligomers, despite any cartoon that you will see in any article. And so the first site where oligomers are formed is in the endoplasmic reticulum, and by expressing a nanobody in the endoplasmic reticulum, we can target them, we can block them there and avoid secretion in the extracellular environment. So we can block the fire of the forest by protecting the neurons inside, and we can show that this is effective in mice models in vivo. So the idea now is that as a milestone of our project in the ecosystem, we are setting up a startup company based on our granted patented technology, based on 25 years of having pioneered this idea of using antibodies as genes. And we believe that this technology can open the way to the world of conformational specific misfolded proteins inside cells, and for what we call a subcellular precise pharmacology. And so, of course, we have an expertise on neurodegeneration. This is where we shall develop this technology, but the value of the technology platform is very well beyond the internal therapeutic focus on central nervous system. So let me thank you for your attention, and my intrabody group at Square Normale, Gabriele Ugolini, Simonetta Lisi, who is really the queen of the libraries, and then PhD students Angela, Rachel Bitonti, Martina Dassolo, and Rajesh Jacob. Thank you very much indeed.
Thank you very much for this fascinating presentation, and we already have a question there.
Amazing presentation, thank you very much. Is it possible to think of nanobody drug conjugates? Did you think of this?
Yes, well, that's a very, very good point. Of course, once you have the core of the nanobody with an exquisite molecular specificity that you can engineer, select, or tailor, and then, of course, we can add either effector functions if you deliver these as genes, as I try to convince you, but also if you deliver them as protein, you can chemically couple with chemical conjugates of interest. So this is definitely around the core of a nanobody that we can derive from our libraries. We are very, very happy to sit down and think about possible conjugates of interest. That is the short answer. Thank you, Claudio, for
comment on that?
And nice presentation, thank you. What about the stability of this nanobody inside the cell? Because this is a very important point. When single-chain FAV are delivered through the blood, they have a very short half-life. In this case, something is changed.
Yes, thank you very much for asking again. This is why we developed intracellular libraries and biological selection. We, around the universe of all possible antibodies in nature, our selection system is selecting a nanobody under conditions of intracellular expression, so we are certain that the nanobody that we select have been selected under conditions of intracellular expression. So we are sure that they are stable enough to work because we have selected a priori those nanobodies under those conditions. That is exactly why we don't use phage display libraries or ribosome display libraries or other libraries or even monoclonal antibody from humans, because we select intracellular nanobodies, our sprint libraries. Thank you.